Okay, very simplified version of cellular respiration. We start with glucose, and glycolysis happens where? In the cytosol, outside of the mitochondria, but still inside the cell. And it's gonna get broken into two pyruvates or pyruvic acid. They're the same thing, and two of them. And in the meanwhile, we're gonna make ATP and NADHs. And these two pyruvates will go into the mitochondria and participate in this something called Krebs cycle. While it's going in, it's going to lose carbon dioxide, and you're going to lose more carbon dioxide during Krebs cycle. And during Krebs cycle, you're making two ATPs as well for one glucose. And all these NADHs and FADH2 that were made in Krebs cycle and this intermediate step called pyruvic oxidation. Don't worry too much about this pyruvic oxidation. I made a detailed video about this. All these NADHs and FADH2, they're going to participate in something called electron transport chain that happens on this inner membrane here of the mitochondria, okay, where it's going to make a whole bunch of ATPs from ADP, okay, during oxidative phosphorylation. And then the electrons from all glucose that were carried by NADH and FADH2, they need to get picked up by oxygen here, and oxygen turns into water. So basically, whole overview of the cellular respiration, you start with glucose, C6H12O6, and then in the presence of oxygen, you're going to make water, and then you're also going to make carbon dioxide, and then a whole bunch of ATP. Okay, so that's the whole point, making energy from glucose. And then that glucose will turn into carbon dioxide, which we breathe out, and oxygen picks up those electrons from glucose and will turn itself into water. So if you want to balance it, except the glucose here, you have to put sixes here. Okay, and you're going to make a lot of ATP. However, if there's no oxygen available, if there's nothing to pick up those electrons, what happens? They're going to go through. Well, before that, let me talk about ETC for a little bit. Okay, all these NADH and FADH2, they bring electrons from glucose basically, and they're going to drop their electrons to become NAD plus and FAD. They're going to go back, okay, and then pick up more electrons and then come back and drop their electrons here. So as electrons go through these proteins that's embedded in the inner membrane, what happens is that oxygen with the addition of H pluses, it's going to pick up electron and become water. Okay? And as electrons go through these, all the protons or H plus, they're going to get pumped into the intermembrane space. So in the meantime, you're going to have a lot of H pluses, okay, all concentrated in intermembrane space. And using that gradient, the only way H plus can go relieve that stress is go through ATP synthase. And it's that power that phosphorylates ADP into ATP. Okay. Now, if there's no oxygen available, there's nothing to pick up those electrons. So all these systems will shut down, except glycolysis. Let's talk about what happens if there's no oxygen. If there's no oxygen available, the end product of the glycolysis, which is 2-pyruvic acid. So if you're animal, you're going to go through lactic acid fermentation. If you're bacteria or yeast, they're going to go through alcoholic fermentation. Okay? So this happens in cytosol as well. So this is called anaerobic respiration because no oxygen is required. Okay, what happens is that as pyruvate turns into ethyl alcohol in alcoholic fermentation, it's going to lose carbon dioxide. Okay, and then if we're an animal, we're going to turn into lactic acid. Now, what is the whole purpose of having fermentation? It's not to make ATP. You don't make ATP here. You don't make ATP in fermentation. Fermentation purpose is to regenerate NAD plus. That's important for glycolysis. You see how NAD plus is needed so that glycolysis can go, all right, so that you can make ATP. That's why the cells go through fermentation if there's no oxygen available. However, if there's oxygen available, what happens is that lactic acid can convert itself back into pyruvic acid, and that pyruvic acid will go into the mitochondria to have Krebs cycle and etc all over again, but not for alcoholic fermentation because they lost carbon dioxide along the way. So even though there's oxygen available, this ethyl alcohol cannot get converted back into pyruvic acid. So this is just one way. However, this is like if there's no oxygen, lactic acid. If there is oxygen, it's gonna go create pyruvic acid again.